Welcome to Elmar's ecosystem.
It's an ecosystem. Everything is connected.
It's an amazing experience.
Hi Naomi, I'm here with Naomi. Yeah, we're here on the SAEF 2 on our way back to South Africa. We had a successful journey, well, you had a successful journey to Marion. I was there for a year. And she met up with me to do some nice weevilling and work with invertebrates and plants. And we just had a lot of fun. And obviously she's busy with a nice PhD on the weevils, am I right? Nee?
Yeah, you're right. I am busy.
And she's going to speak to me about it, and we're going to have a lekker conversation. So yeah, my first question is, before we get to that, I actually want to find out by you, like, how did you get into weevils, or specifically? That's my first question.
Okay. So, I've had an interest in environmental science for many, many, many years. And I worked in environmental management. And one of the things that, or one of the animals that I worked with was pygmy possum. And the pygmy possum was under threat because the moth that their food source moth, the bogong moth, was in depletion. And I decided I wanted to monitor this moth. And so I went out and tried to find the best methodology to monitor the moth as part of my work. And that got me hooked on insects. After that, I was like, I just want to do insect research.
And what part of Australia is that?
So in the alpine areas.
Alpine area.
Yeah.
Okay.
Yep. So it was a mountain pygmy possum that eats the bogong moth that migrates up there. And yeah, working with the moth was so much fun. And then I just decided I wanted to work with invertebrates in general. And I did some stream monitoring and worked with macroinvertebrates in the streams. That was really cool because in the alpine streams there's some amazing, unique creatures.
Like stoneflies and all that.
There's a stonefly that, lives 3 years as a larvae and then emerges as this massive adult, like the size of your thumb.
Wow. And
hangs out for a few weeks, mates, and then that's the end of its life cycle. Yeah. Like, they're phenomenal. They're hardy. They're amazing.
Yeah, that's cool.
So working with that, and just after I finished my honours on macroinvertebrates, I was looking for the next thing, and I knew I wanted to work with cold climates. So alpine, but I really wanted to do sub-Antarctic stuff because I had a fascination with, or I have a fascination with sub-Antarctic and the Antarctic region.
That's interesting. I thought, like, as an Australian, you would want some heat and not cold environments.
No, colder the better.
Okay.
Cool things happen with the cold.
I have to say it is like that. I also enjoyed the cold, but I missed the warmth, I have to say.
I can imagine you would have after a year.
But I think I'm going to miss the cold now.
It's in your bones now.
Yeah, it's in my bones.
Yeah.
But anyway, sorry for interrupting.
And so, that when there was a job, a PhD advertised, and it suggested weevils, apart from other invertebrates, and I was hooked. I thought, that's perfect.
So
that's what I want to do.
You didn't meet, like, Stephen beforehand? It was a application that you had to... Yeah. Okay, that's cool.
Yeah.
How interesting is that?
Yeah, and it's been so fascinating, and I've loved everything I've learned so far, and I know there's so much more to learn. Every day there's something new, right?
That's what's so cool about life. You can just learn everything. That's the whole point of living for me, just to learn more things. But
I think that's the thing with the weevils, right? So there's heaps of work being done on the weevils, especially the southern Indian Ocean islands. And when I first started, I was like, what is there to, like, for me to add? Like, what can I add to this? Because there's so much already written. But then the more you start, like, learning and questioning, the more you realize, there's still heaps we don't know. There's always heaps we don't know.
Yeah, but it's like when you arrived, you just showed me some stuff that I didn't know. Like, and instantly I'm like, Oh man, if Naomi was here last year, it might have been better for me. But like, that's how you learn, you know.
Yeah.
You can't always have all the information ready for you.
No,
It's like a process.
it is absolutely a process. And
Because-
you learn through doing, right? Like,
Mm.
having read all this stuff about Marion Island and the weevils, I thought, yeah, I've got this. I know where I have to look. I know what I have to do. But even just those little like
Yes.
moments when you're in the field and you're looking, you know, in the Azrella and you realize, oh no, okay, that's, you know, this is how they tuck themselves in. It's like little tiny things that you can't learn from a book.
Yeah.
You have to be in the field and you realize,
Yeah, you can't
little
learn.
behaviors that,
Kinders read as many papers and books as you like. You still don't really know.
Nah.
That's what I like about field work. That's the whole reason I enjoy it so much, because you learn a lot more.
yeah,
A lot more than just looking at some photos. And
yeah,
I don't feel that's useful. It's a combination. You need a combination of both
it's
to
true.
learn more about it.
And you're very good at finding them in the field. Finding any invertebrate in the field.
Yeah, but
It was
it's
super fun.
well, I've done it for a year, so
I know, but you know, it doesn't necessarily mean you're gonna have the eye to.
yeah, true. But I think I struggled at the start, if I'll be honest. But I can remember the first time I didn't really understand where everything fits in. Like before you came, I knew there had to be new niches. There had to be niches for beetles, because I was unaware of that, because I didn't really know their biology that well. Because I didn't have a specific interest in the invertebrate per se. I just wanted to try and find as many different ones I can,
Yeah.
because that was pretty much what my protocol is. I need to find as many as I can and collect them, you know.
Yeah.
So I just looked everywhere. I just looked like under the moss, in the soil, in the azo, on the rocks, on the lichen, you know, etc., etc. And that's how you like see cues, and you see different ways of their habits and where they hide, and you learn, pick up on stuff. Like I can remember, you know, that elongatus we found. I always found their larvae up at Cathedral, because I look under that Andrea moss and I lifted that, and I always find the larvae. Now, when you came and you told me the elongatus is there, now it gave me like a,
Yeah.
it's like a, I don't know how to say it, but like it gave me like a clue. Oh, so that must have been,
It connected.
it
the
connected,
dots.
it connected the dots, yes.
Yeah.
And it connected. Oh, that larvae I found there all the time was probably elongatus larvae.
Yeah.
So that's how you like connect the dots. But at the time, you're not sure where it fits in. And that's what I feel like. Combining people's knowledge together and doing research is really important.
Yeah.
And being on your own and going to do it yourself is actually could be good, but I feel like you always need to collaborate with a lot of people, because then we're gonna find more answers like that. So yeah.
Yeah. You spot more as well. You'd start to. I remember when we were looking for in the Cookie Eye at near Macaroni Bay area, and we were looking. I was so busy looking for the weevils, and then you were like, "Oh, there's so many of this Colembala here." It was like, "Oh yeah," like I hadn't even like they were there. I just hadn't
You hadn't noticed
focused because I'd been focused.
Yuck, there's a lot of Colembala.
Yeah,
Yuck,
yeah,
especially
and then
there.
you can't help but see it.
Yeah, you can't help to see it. It's like some of the spots with the cotula. I actually saw that name change now. It's not cotula, they call it leptinella now.
Okay,
That's what happens when you get good internet. But that leptinella, there's so many aphids there. Like sometimes in the summer and in the winter, I should actually have taken more note on that, but I didn't. You know, you just go and you just work. It would have been interesting to see. But anyway, getting ahead of myself now. There was just so many aphids, just like crawling over each other, like millions, like just on this cotula, on the leaves, on the flowers, just everywhere. And you're like, yo, what's going on here?
Wow.
Like I puttered them, but it's like I just stopped after a while, because it's like, how many should I take here? You know, I would have had like flipping thousands of them in a putter. So I was like, yeah, I'm not gonna.
And
put
had
all
you?
of them.
Did you ever see that again, or was that like a one
Yeah,
time
that's
boom?
the thing. It's not like all the time. It's
No.
like sometimes there's like booms. I think there's booms. Like
Yeah.
sometimes there's a lot, and then you come the next day or in a week, you'll find nothing, or you'll find one or two.
Yeah.
That's the cool thing. I hope like Abdul will also notice these things, and
Yeah.
he'll enjoy it a lot. I think he'll enjoy it a lot.
And I guess you don't really appreciate them at those moments at the time.
No, you don't, because you don't
'Cause you
realize
just see it and you go, oh, that's
Oh,
weird.
that's
there's
probably
a lot.
normal. That's
Yeah.
maybe normal. And then you come there in a month, you're like, oh, but they're not here anymore.
Yeah.
It's the same with the weevils, though. Sometimes some days, like those misty days, and it's quite semi-warm, like 8 degrees or 10,
Yeah.
or almost 10. Let's say almost 10, and it's just misty. You'll see a lot.
Yeah.
On top of the azure, all the ectomorhinus on there. Then I saw the rock ones, the parvulus
Yeah.
on the rocks as well. I think that's the two that I mainly got in my collections.
Yeah.
Just by looking how you did it now and the IDing, I think obviously those two are the most common ones, my transects, because I didn't really have transects right on the coast for
Yeah, you
the
had a lot of
Ballerines,
similars.
Ethanol.
Yeah.
Yeah. But yeah, it's very interesting. Some weather, just you can see there's just more. And some spots have more. And
Yeah.
some other spots are dead. There's nothing. You're like, why?
Or appearing dead.
Or appearing dead, yeah.
Yeah.
Maybe you just
You gotta be
you're,
there at the
you're
right
down the
time.
right on the wrong day, exactly.
Yeah.
So yeah.
If only you could sit in a spot forever
Mm,
and just watch.
yeah. but
Imagine.
it's for every species there, like for the birds as well. You know? It would be nice to sit there for the whole day and just observe.
Yeah.
But now you have like work to do and you can't really sit there the whole day and observe. People are gonna get angry. Or you're gonna get cold as well.
You
So.
get cold, that's for sure.
But yeah, it was a great experience.
Yeah.
Yeah.
Tell me what you learnt or know about the flightless moth, because when we were there, we saw a couple of adults, maybe
But you
4
know,
or 5.
There's actually, I'm not sure. I don't know as much. Only thing I know is that there's actually maybe 3 species and not 2.
Okay.
So there's that one is the Pringlia species, and then there's another Pringlia species, perhaps. I'm not sure. This is things I didn't have information on before I came. Didn't do my research that well on them. I didn't really have time to read a lot, but I think I should go read. But I observed it in the field when I collected it, that some of these Pringlias don't look the same.
Okay.
So I think there's a different species there. I think there's a Marioni species. I looked at the literature a bit, and then there's another name. There's the other species. But I'm not sure what his name is now.
Okay.
But I don't know if it has to be looked at again or anything. But then the other species is the Embryonopsis species. This is a more wider in birth. His wings are more bigger and it looks thicker and it's tinier. And I saw that one a lot on the cabbage, because the cabbage.
Okay, I was going to ask you, did you find them like one more on one habitat or
Yes,
location?
so that
right?
one Embryonopsis I know is the larva eats Paucocchii, but sometimes I find the adults on the cabbage flower. That's what happens. I'm not sure if I don't think they can feed though.
No, they're
I think
potentially not.
Yeah,
Like,
they're not
often
supposed to
adult
be able
moths
to
don't.
feed.
Yeah.
No, I don't think they feed, but maybe it's like a area they go to find each other, or I don't know.
Does the cabbage have flowers at that time? Could
Yes.
they be nectar feeding?
They might be, but I don't think so, because I
Okay.
read about it that they don't feed.
Okay.
The flightless moths. The adults don't have feeding structures. They just live for 17 days,
Oh, literally.
and they just walk around and try to find a mate, and they die. They lay the eggs and they die.
Okay.
But it will be interesting to look at that. The other thing I know about them is the larvae is so generalist
Oh, really?
species, because they eat anything, I think, because they're decomposers. I find them under liverworts. I find them under albatross, the nests, the old
Yeah.
nests that
Yeah.
clutter. Sometimes I find a lot under them. I think it's just the energy, obviously, in the soil, with all
Yeah.
the poop and all of that. And then you find them under Paucocchia. You find them under
Yes.
polypogon roots. You find them under sometimes just rocks. This one time at Cathedral, There's nothing. There's no moss in the area. I can show you the photos.
Yeah, what's it
maybe.
eating up there?
There's no moss. Okay, you know those small moss I talked about. There's some small moss species there.
Yeah.
But sometimes I lifted rocks there, and there's just like nothing underneath. Not even lichen. It's like bare soil, and the soil is like gravelly and it's like dusty.
Yeah.
There's some larvae there. I'm like, huh? Where do you come from? Why are you here? So how long can they stay like dormant maybe?
Yeah.
And just move around and not have to eat? Maybe that's why they live so long. Maybe they don't need as many energy if they just stay in the cold. You don't need to do much, right? You just chill. And then when you get hungry, maybe in a month they'll go eat.
Those ones at a high elevation, it sounds really interesting.
So there's a lot of stuff you can do. Like, that's the cool thing. Especially with the invertebrates, I feel like you can do a lot of physiology studies on them. There's a lot of gaps there.
It would be really interesting to raise those.
This is
and
the thing.
see which one that comes out at. And
So
whether or not they've got the same.
these are
physical
things I need to like write down. Just stuff that I observed and send it to Michelle. But she's just aware of it because she's not coming to the island recently. She has a lot of stuff and.
But yeah, those little things like that comment. That's a huge potential question, right?
Yes, exactly. No.
Like, is that another species?
Is that another species? Is that
a variant?
A variant? A
is
subspecies?
Is it ones that aren't going to survive, or are they so tough that they do survive?
And then the other question is maybe, like, I know from the records, this feels like it's my podcast now, but it's okay.
It's a shared.
It's a shared.
We're sharing this experience.
Okay.
Yeah.
Because Charlene was at Prince Edward, so she told me when you walk there, and Mashudu actually here as well, of Makudu, what's his name? Not Mashudu, Makudu, sorry. He was also there, and they like always told me like, when you walk there, it's just moths everywhere. Like you'll see an adult running there, and you walk like 5 meters, you see another adult. And then but here it's not like that. It's like you saw. We only found moths.
A handful of times.
Handful of times. And that's how my year also went. Like I did my invert searches, and I was happy when I found a moth. I was like, yeah.
I think I've got a photo of every one, so that's how like it wasn't that crazy many that
exactly that you don't want.
Could actually take a photo of every single moment.
Exactly.
Yeah.
It's not like the weevils, like the ectomoranias. There's quite a lot.
Yeah.
Compared to the moths that you find.
Compared to the moths. Yeah.
So I'll say I found it like once a month, but anyway, like if you compare the numbers, there's a lot there and there's no mice. Now it would be interesting to collect some of them and compare it with obviously the ones on Marion, because that will maybe give you an idea, because I have this theory, perhaps the mice only feed on the one species, or the mice, some of them are, I don't know, more elusive and can avoid potentially the mice from becoming the prey or something like that. But yeah, there's a lot of stuff and work that can be done, and there's a cool thing that you can compare that island with the other one. And that's a cool study that you can do in the numbers, and you should actually also measure them and weigh them. So that's what I did for my one project I did for the soil course. The whole point was to find these moth larvae.
Okay.
But I recommended for Michelle we should have done active searching. But the problem is, it's a previous study that they did in the 70s and in the 80s and the 2000s.
Yeah.
And soil cores were perfect for this because there were more moth larvae.
Yeah.
So they found a lot. And now I'm doing a soil core. I'm lucky if I found one.
Huh.
So
it's quite telling then.
It's telling.
Yeah.
It's totally telling. The mice are definitely feeding on them.
Yeah.
Yeah. And they're feeding on all the plants. Like, I don't know if you've noticed, but most of the plant heads, seed heads,
off. Yeah.
Off.
Yeah.
Cut it off.
I remember someone telling me that, like, I'll hardly ever see a poa cookie eye seed head. And that's why every time I saw one, I was like, Elmar, look!
There's a poa cookie eye!
There's a poa cookie eye! This one has seed heads! And you're like, yeah, they've got that. I'm like, no, this one's got it. I think that's why I was so excited, because I'd been told that I was not going to even see that.
Yeah.
And apparently the Ectemna highness does like to get into the
No, they
seahead.
do. They love it.
Yeah.
I've seen that as well.
But yeah, like it was so few and far between the seed heads that I didn't see any.
And it's that
on this trip.
Embryonopsis larvae. They also eat the seed.
Okay.
Yeah, they feed on it.
Okay.
And that's a different one that you need a real eye for. Like, you think I'm good finding the stuff, but that one is difficult to find.
Yeah, right.
You need a real eye, because they're small. They're really tiny.
Yeah.
Tiny larvae.
And quite still, I guess.
And quite still, yeah.
Yeah.
So you need
the
to
movement.
be patient in the field to find them.
Yeah.
I can remember just when I started, I think it was in June, that's when I started looking at the cabbage just for interest. And I found 2 moth larvae on the cabbage. And it wasn't the flightless moths. There's one moth, Plutella,
Oh, yes.
that occurs. And I found that larva, but I've never seen an adult. I think it's a nocturnal moth that flies at nighttime. So you probably need to do a light trap or something to find them. I'm not sure, but I was thinking, because I found it so early, it's okay, I'll find more. you know, you think you're going to find more, and I didn't realize how rare event that was.
Yeah, right.
But I have the collection, I have it in the lab there, so...
They'd be highly seasonal, I would think.
Probably.
These moths. Like, moths tend to be really seasonal.
Yeah. Just to go back to your question about the moths, like the moths are more active now, early winter and then late summer. Definitely. If you look at my results, like in springtime, I don't find a lot. It's weird.
Did you ever find adults without finding larvae, or was there always an overlap?
There's always an overlap.
Yeah, interesting.
Always.
It's a bit like
it's
weevils.
always you find larvae or no adults, or you find adults and larvae. I've never found only adults and not larvae.
Okay.
That makes sense.
Yeah.
Unless if you're not gonna look under things, then obviously the bias is you're going to find just adults, because adults are running on the surface. While the larvae, you need to roll things over and look through vegetation.
Yeah.
So yeah.
But yeah, a bit like weevils, you can find both.
Yeah.
I'm guessing because you did in your winter samples.
Yeah, I found a lot of larvae.
Larvae and adults.
And adults. Yeah.
I was surprised by how many adults you were encountering in winter.
Really?
Actually,
I think I found over 1,000 probably in total.
There's a lot for winter.
Yeah.
Yeah.
Yeah. No, I
were they very active in winter? Like, did you go out when it was snowy? Like.
Yeah, sometimes I had to. Like, the problem was I was working with Monique, so we stayed for 3 days, let's say for instance at Repetto's, right? And our first day is just setting out the traps.
Yeah.
And then the next day might be a crap day. You know, it might rain totally, but we have to go out and check the traps for mice. And then I have to plan okay, sometimes if the weather's really bad, it's totally raining, pouring, and I would say, Monique, we'll not do it in bird searches today, we'll do it tomorrow. Usually I check the weather and see. So usually I pick the best weather day out of those 3. But yes, there were times that it's bad. I went out. The first time I cut the drawl, and then in September I cut the drawl as well. It was snowy. But I found weevils. I found them in the snow actually, but not as many.
On the snow?
Yeah, some of them on the snow.
Yeah, okay.
Yeah.
Interesting.
But your hair was cold. That's not fun. Your hands get so cold.
Oh, I can imagine.
Because I try to do the same method, you try to look under the roots of things and inside a cavern or whatever. Then your hands get into the snow and then you're like, oh my hands.
Cold and wet. And,
then you just go like this.
yeah. Rubbing them to try and keep warm. Absolutely.
But yeah, that was fun actually. I'm gonna miss it actually. I wanna go back to South Africa and go find insects.
Yeah, well you've got the skills.
Yeah, it's fun.
Yeah. Yeah. I was surprised. What else did you find in winter when you were just looking in the snow?
Not many things. I try to pooter things as well. I find the invasive Columbella in winter.
Really? So that was still active in winter?
Yeah, but the thing
didn't
is,
seem bothered.
Everything's still active in winter.
Was it on the snow or was it under the snow where it was
under the
a
snow?
A bit insulated.
Yeah. So you won't find Columbella on top of the snow. No. And you have to find patches where you can get in under the snow or under a rock, for instance. Or lift a rock underneath the snow But usually, it's obviously the snow is not like knee deep. It's like when the snow is just a bit covered, like let's say
Yeah.
5 centimeters or something.
Yeah. Okay.
Like the one time it was like knee deep, then I just thought, nah, we're not doing it now, and I just skipped it on.
Yeah.
Obviously, it's
Yeah.
just a waste.
Yeah.
Yeah.
I think there's a Columbola that does go on top of snow in like Norway. I think they call it like snow fleas or something.
Snow
like,
fleas.
Yeah, and they congregate on the snow.
Okay.
But I think it's like towards the end of the season. I think it's I don't know, perhaps to
Yeah.
mate and gather kind of
Yeah.
as a big group.
But it's like, I'm glad Abdul is replacing me because he also has knowledge on that. Like,
He has a lot of knowledge on that.
yeah. So, like he told me when he saw Cathedral, he told me, and I was really impressed by him because he said, I know what we're gonna find here, and I'm like, okay, what are we gonna find here? We're gonna find blind Columbola, and we're gonna find mites here.
Yeah.
And they were like, well done. That's exactly what you find here.
Yeah. Nice.
Like that's what I found on my soil course. You don't really find anything else there. And obviously active searching you can find some weevils and that, but in the soil core you're only going to find mites and Columbola, blind Columbola specifically, because
Yeah.
they live under the soil.
For a lot of the year.
Yeah.
Or for all of the year.
So there's 2 species on Marion. There's Macromesia and then Tolbergia. They're the 2 blind ones, and they're very easy to identify because the one does not have a furca, the Tolbergia, and then the Macromesia has a furca. So it's very easy. If he's blind, he has a furca, that species. If he's blind and he doesn't have, that species.
It makes sense, like a soil-dwelling one, to have no furca, because
why would you want it?
Flick away from nothing?
Well, there's nothing to flick away
How
from.
are you going to do that? But the one that's got the furca is
And it's
interesting
blind.
because it's blind. So
Weird, man.
yeah, it hasn't lost.
It hasn't lost it yet.
Yeah.
So it's probably semi in the soil underneath and semi not. Sometimes maybe it comes out, maybe it goes back.
Yeah, it's kind of a little clue.
That's probably the niche.
Yeah. Otherwise,
that's how
they'd
you
be
pick up
the
on
same.
these clues, right? It's very interesting. And like the invasive ones, mainly at like obviously the Ceratophycella is the cosmopolitan one, and it just gets to all the, obviously where all the nitrogen is, all the seal crap and the penguin poop, all the biotic sites. Millions of them. It's crazy. Just so many.
But you've never found it in a poor site. Like
Never. I never find it like in a poor habitat. But now there's another interesting thing. I know we shouldn't really talk about Columbola now, but that's where the conversation went to now. But
they're still part of the group.
this is the thing that baffles me. It's a Columbola, right? How does it move around? It jumps around with its furca, whatever, or it takes mode by flight from the birds, or maybe it can move like that, or from and catches in the wind. Now you get these spots like where the albatross poop, and it's very far away from a biotic site. You'll have an albatross, a wandering albatross, high up in the mountains. Now I check through the Pyokukia out there, and you find an invasive Columbola. And you're like, how did it get here? It's like, there's a lot of cool questions you can ask, and I think that's like something Abdul would really like.
Yeah.
Doing a study like that.
Yeah.
And he should investigate that. The only thing is we need to write the protocol and all of that, and he has obviously other work to do.
He's got a lot going on.
That's the problem.
So many cool questions though.
Yeah.
Because like genetic work, you could do amazing stuff with
Exactly.
that.
Genetic work.
Sample.
I'm not
those
good at genetics.
high sites.
I did some genetic work when I was doing my masters actually on fungi, and it's pathogenic fungi. I worked on bark beetles and ambrosia beetles.
Oh yeah.
How they kill the trees. I was just looking like at Rosarium and how they kill the trees. I was just looking like which species is it. I didn't do any like comparison or whatever. I just needed to figure out what fungi it is.
What it was,
That's it. Yeah. But otherwise, I don't know how to do. I'm not really good at genetic work. But yeah, it's like following a recipe. When you do some genetic work, PCR and all of that.
Yeah, I'm sure once you have a protocol
Yeah, it's
and
not
you've
that difficult.
got the research
But it's quite
questions.
stressful doing that work. I don't know if you've done it before.
I think the stressful part about anything to do with genetics is the cross-contamination.
Exactly.
You're constantly worried
You're constantly
touch
worried.
something that's then going to touch
That
the
just freaks me
specimen
out.
that's-
I can't take that. Like,
and it's all invisible.
Yeah.
So you can't trace it visually what you've done. You need to just be very conscious of
but
every
it's like
single
the
step you
primers
take.
you
Right?
use for animals like insects. Those ones it was so difficult, but that's where the contamination gets, because your skin obviously can contaminate those ones. But the fungi is better because it can't pick it up.
Yeah.
At least. But then you get obviously mold and stuff that might be in your clothing.
Yeah.
And you don't know. So that's
Yeah.
what just freaks me out. It was stressful, that for me.
There's so many potential cross-contaminants.
Yeah.
Yeah, it's true. It is stressful.
So genetic work's cool, but I would just want to give it to someone else to do
Yeah.
rather. I don't want to do it.
It tells cool stories.
Yeah, it tells cool
if you
stories.
can sample in the right way.
Yes. I just
Yeah.
like the evolution and the history of where things come from and why do they move and how do they move.
Yeah.
That's what fascinates me in nature.
Yeah.
I love that. I love how things adapt to their environment, you know. All the microhabitats,
Yeah.
the
The
niches, they
niches
fill.
that they find over time.
That's what makes invertebrates so interesting, because
Yeah.
they're smaller, so their niches, you know, their environment's a bit smaller, and we
Yeah.
can't always see that. As humans, we just see this one habitat. But for them, there's millions of habitats maybe. Who knows what
So
habitat?
much variation.
We don't even
right?
know.
Yeah.
There might
Like
be some bacteria spot there. There might be some weird mycelium growing under the soil that we don't even
Yeah.
aware about.
Yeah.
You know, so that's what's fascinating.
That's true. It's true.
Micro ecosystem.
Yeah.
Yeah, it's very cool.
Yeah, it's like mind-boggling when you think about like the time that's elapsed to create all these little niches.
Exactly.
But I think that's why it's cool with invertebrates is they've got such short generation times that it can happen relatively quickly compared to a lot of things.
A lot of things, yeah.
Yeah,
That's what makes
Yeah.
it so cool. Yeah.
which is why it'd be interesting for the invasive stuff.
Mm.
How quickly are they adapting and changing?
But yeah, I could see you enjoyed this trip a lot. This
I've had a
Marion.
great trip.
Yeah.
And I really enjoyed working in the field with the whole team, really.
Yeah, it was great.
Even going out with non-invertebrate teams,
Yes.
I learned so much.
Yeah, you learn a lot.
About just the environment and thinking about the environment differently.
I can remember at the start how afraid you guys were to go out. Oh, is there gonna be Myers? I was like, don't worry about that. I don't know why people just talk a lot of crap.
You hear a lot about the Myers.
Yeah.
Yeah, I think
They
they make
just
good stories.
Make these. Yeah, they make good stories.
Do you know I successfully didn't get Myred?
I know. I never saw you got Myers.
No.
That's amazing.
No.
You're probably the only
quite
one.
proud of that.
Yeah, you
I'll
should be
wear that
proud.
as a badge.
Yeah, you should wear it as a badge.
We need to create
Didn't
a
get
sticker. I
mired.
did not get mired on Marion.
We should
Be
actually
proud
do
of
that.
it.
You're probably the only one. Yo, no. I got mired a lot of
I
times.
can imagine.
I even got mired the last day of walking because I was.
Did you?
Yeah, just for fun. I went over this massive mire that I knew about at just behind Juniors, there close to Tafelberg actually. It's this
when you went out that
floodplain.
way.
Yeah, it's this floodplain section. I was like, let's take the risk. But yo, that is hectic. That is not even
How deep?
No, I just went to my knees. But
Okay.
you can go really deep. You can
Yeah.
go swimming there. So if you have to be careful. Like if you give too much weight, then you're gone.
Well, there would have been a lot of rain and a lot of snow, so I can imagine that
Yeah.
had the ability to be quite deep.
Yeah, it's amazing how you were like a small child and you know those books you read about quicksand and
Yes.
you're like, that will never happen. Where's quicksand? And then you come to Marion.
It is like
There's
Marion's
wallows
version of
and
quicksand,
mires.
isn't it?
Yeah, basically. It's hilarious.
Or you take a step near a wallow and it
And
just
then,
like it
whoa.
all just moves like a
Yeah.
jelly pillow, and you
Whoa.
think, okay, I'm gonna step off this now.
Yeah, and you always think like, when is this thing gonna pop?
Yeah,
You don't
exactly.
know when it's gonna pop.
Exactly.
Yeah.
Exactly.
Yeah.
Yeah. Super fun.
But yeah. Oh, I think that answers your question. But I don't know. I can't even remember what your question was. But yeah, what do you think is your objectives through this study now with the weevils?
So with the weevils, my big goal is to continue this long-term temporal data set that we've got on Marion Island, but we also have some from Prince Edward. So you're talking about Prince Edward bit.
Yes.
And so some of my study will do some comparative work because Prince Edward doesn't have the mice, right? So there's some really interesting things.
It might be
that we
larger
can
there.
Yeah, and work done previously 10 years ago, 15 years ago, definitely found that the mice were altering the body size, and we know that even with temperature they respond to changes in temperature with changes in their body size. So in colder areas they're different, and it does depend on whether or not there's the predation pressure of mice as to how they respond. So my big goal is to take these specimens away and work with them, measure all of the body size and other trait information that I'm really interested in looking at, and then trying to see if we can piece together what's changing over time in response to the invasion of the mice and also climate pressures, and see if there's any other pressures that I can feed into the models to see if there's anything else that comes in, because whilst they're the two big things that we as humans think of, like you said, there's like microhabitats, there's things happening.
It's difficult to put that in, to incorporate that. That's the problem.
But I think precipitation's another thing I really want to put in, because we know Marion is getting drier, and apparently the Mayas aren't as bad as they used to be, I hear.
That's what they say.
And so, you know, factoring that change in precipitation in that could be important as well. But yeah, I think trying to put into the models as much as I can that seems realistic, that's manageable, and finding out what's driving these changes in the way of, you know, historically and contemporary and right now. And then once I've got that, I'm going to use it to put into the perfect model, the best fitting model, the predicted stuff for the future. So what's predicted in the way of climate, precipitation, invasion risk, all the predictions that seem relevant for the future that we've sort of, you know, are forecasting on the trajectory that we're on right now. Put that into the model and see, okay, well, what does that mean for terrestrial biodiversity on these amazing Southern Ocean islands? Are we going to, what are we going to see? What's going to happen? And try and predict that, because then if we can predict that a little bit, we can inform conservation and go, you know what, this species here that we've seen hardly any of since the 1980s, if we keep on this path, the model's suggesting it's going to get itself into a genetic bottleneck and disappear. Or, you know, maybe it's like, oh, we've found this species is adapting really, really well to this particular pressure. So maybe it means that there's always winners and losers. There's winners and losers in everything with ecology and environmental stuff. So trying to figure it out before it happens can really help, because then you know what you need to champion for in conservation. You know what, you know, policies need to happen, or protections need to happen, or, you know, species need to be put on an IUCN red list to get attention so that they don't disappear off the face of the planet. This science is how you do it, right? Science is how you put numbers to things that you know matter. I can remember when I started, like, I was also looking at the altitudinal gradients, because some of my transects are very high up, some of them are middle, and some of them are low elevation. Like if you're talking about body size, you can actually take my weevils, you can just take ectomorhinus, for instance, at all the sites and just measure them. See if they actually... And there has been work done in the past that's shown these altitudinal gradients, and that the gradients change and reverse when the predation pressure of mice is added, compared to that on Prince Edward Island. But that said, it was a long time ago that last lot of work's been done. So doing it with this new collection, I think it will be very cool. I think it will be really interesting to see. And the mice numbers have increased, definitely. Absolutely. Yeah. It's weird, like at our takeover, the mice felt more than this takeover that we just had. Okay. Like,
don't know if it's because I got used to them but it felt like there were a lot more. Like around the huts and around base. Maybe we were just a cleaner team or something. I'm not sure. But it just felt like there were less this year, but I'm not sure. Obviously, it might not be the truth. And everyone in the team were like saying, yeah, it's because you killed a lot. But I don't know if that's the truth. But
it's interesting because, like I think you mentioned that there was a lot last takeover, even inside,
inside the base.
Inside buildings. And then this year not so, but then when we had that snow event, that was the first time I actually saw mice outside the day, during the day. I was like,
they don't have a choice.
They're hungry.
They're hungry.
Yeah, and that was a bit telling that when there's a pressure, that's when they give in, really.
They give in.
They become more visible.
It's the same thing that I think I told you at the repetitors that happened. I captured mice there at night time. It snowed overnight. And then all the mice came out and ate their brothers that I caught, and I feasted on them because there was nothing to eat, obviously, because it was covered with snow.
Wow.
So they just go eat anything they can.
Whatever food source they can.
They can. It's crazy.
Yeah.
So they're very adaptable, the mammals, the rodents especially. And you mentioned something now with the beetles being adaptable. Do you think invertebrates can be as fast as mammals, maybe?
Well, that's a good question, hey?
That's
like
a question I want to figure out. Like, imagine they can, and we're not aware of it.
Yeah. Well, that's true. So part of one of my research questions is looking at not just the trait of body size, but looking at the coloration.
Yeah.
And I think that will be really interesting to see.
Yes.
How that plays out with the predation pressure of mice and knowing what they can see, whether or not like you suggest, the weevils are adapting, evolving to the pressure. Or
maybe not at all.
Or perhaps not at all.
Not at all. They're just going on with their business, not even realizing they're being killed.
Perhaps it's selectively not a strong enough pressure that can be genetically passed on as quickly as it needs to be.
But you probably also notice, like, sometimes when you search, you find there's light rodges lying on the surface. Do you think that's mice? Because I think that's mice that eats them and then they leave the elytra or they leave a part.
Yeah, I definitely was thinking that.
That was
because you'd often find them near mouth, like there'd be mouse scats nearby.
There would be scats.
Yeah.
That's what I was thinking.
Yeah.
And you found it a lot in the Baltic sites, because I think the mice are really abundant in Baltic sites. Because there's a lot of food,
a lot more
poop,
food.
A lot of invertebrates, a lot of plants, seabeds, and all of that.
I don't know enough about how mice eat, and it's something I probably should talk to some of the mouse-free people with about, because like I'm wondering, like, some small mammals, they physically use their paws to sort of manipulate the food in their mouth and sort of will roll it around and sort of take the softer parts out of a fruit or a seed or an abdomen of an insect, and then leave the harder outer bits.
Yes.
And that's why you see little piles for certain mammals. But I don't know how much mice do that, or if they just put the whole thing in their mouth and crunch it away and a piece falls off. Or are they selectively actually preparing their meal in a way?
But like in my year, I even saw a skua eat weevils. Like I saw one poke.
Did you?
I did. No, and paddies. Like we know about the paddies.
Well, we know the paddies love them.
But I've even seen skua like picking on the azo. Then I saw one go into his mouth. I'm like, whoa.
That's interesting.
Yeah. So.
That's cool. I did not know that.
I can't actually believe that thing can give him any energy though. It's so small. Maybe it's just like eating a popcorn for the school.
Just needed a snack.
It just needed a snack, or just playing around. It's interesting.
Huh. Yeah. That's really cool.
Yeah.
How much have you seen of the paddies eating them?
Not as much. It's in the winter. Then they come inland.
And you see them come inland?
Yeah, and the gulls. Gulls and the paddies.
Okay.
When it's winter, they come inland.
And how do they forage? Like, are they focusing like on azo, or are they looking like, how do they forage?
They just forage like randomly. I don't know if you've seen them on the coast foraging, like between penguins. It just feels like
random pecking.
Yeah, they just peck random.
Yeah.
It's the same inland. Oh. It's like just this random pecking, and then look under a rock or something, pack there, and then they'll go into a grassy pack. They're very weird birds, actually. It's like they don't have a process of finding food. It's like they just peck everywhere.
Yeah, it seems
They just peck everywhere. Okay, if we peck here and we peck there, we're gonna have enough food tonight. It's like that.
It is like that. It seems so unsystematic.
Yeah,
I'm sure it works
it's like they
for
don't
them,
have a
like
process, a thought process when they forage.
Yeah, it seems to work, but it looks bizarre.
Yes,
Yeah.
it's hilarious.
Yeah.
Actually, it's very funny birds. But they pack anything, as you've noticed. They pack your boots, they pack your clothing.
Yeah.
And like I'll put out for the seed counts, like what happened to Nicola, where you put your PVC pipes, your quadrant. Woo! They always can pack that away and play with your camera, or you sit and have lunch. There will always a paddy will come sit with you, or a skua sometimes as well. Then I had like kelp gull also following me, because I had that red, I think I told you, I had that red strap of my camera hanging out.
Oh.
And they see the red, and then one follows you, and then another one, and then later there was like 20 kelp gull around me. I'm like, what's going on?
Really?
And then I like realized
What
that's
about the
red.
red?
And I hid the red in my pocket, and then like 2 minutes later, all of them just scattered again.
What do they eat that's red normally?
It's
What-
for
what?
blood. It's like
Oh,
for
it's
anything,
blood.
for
Of
bleeding.
course.
So they're looking for anything that's
They're
died.
attuned to red.
Like they
Huh.
eat the mice. Me and Monique, usually when she's done with the mice, or it's the mice that she doesn't need to use, we caught too many, or like she didn't collect all the mice we caught, because we caught a load. We don't need as many.
There's-
So we just went to throw them down the chute at Crane Point into the ocean. What else can we do with them? And then all the gulls will come, whee, fly in, choop,
Oh,
eat the
wow.
one, choop, choop, and they will
Wow.
just eat all those mice we threw out.
Interesting.
So yeah, that's a problem with the eradication. I didn't actually bring it up in that seminar today.
It's an interesting
I wanted to
thought,
ask
isn't it?
them, but because when they going to throw the bait and get like the toxins in, the gulls and those things are going to eat the mice, and they're probably going to die. They're going to get the toxins inside of them. So that's something they know about, obviously. But I wanted to ask if they're going to do the same at Goff, where they capture birds and keep them in captivity because they might die, like the baddies, for instance. But I don't think they'll do that because it's too much money at the end of the day. So the problem is some of the birds are going to die probably of toxin, the
Yeah,
poison.
it'd be interesting to have a chat, but I don't know enough about it.
But yeah, they know about it, so they'll sort it out hopefully. But yeah, it's very cool. It's a cool island.
Very cool island.
But I think you should go to more. Seriously.
I like that idea, and I think I should go to more too. I would love to see so many, because I've been reading, just about the weevils on every single island.
Yeah. Aren't there some Australian islands that Australia owns, or what's the word?
Yeah, the Australian territories.
Yeah, territories, yeah.
So Heard and McDonald Island, not too far from here,
Yeah.
really, in the scheme of things. And then Macquarie Island's on the other side of Australia.
So
towards
that's
New
Australian
Zealand.
islands, well.
And that one is as well. But
Okay.
Macquarie has no weevils. I remember telling
No
you
weevils.
this.
Yeah,
No
you told
weevils,
me. Yes.
which is fascinating.
That's so
but
weird.
frustrating for me, because I would love to visit Macquarie Island.
Yeah.
But
I think
yeah,
there's like orchids and stuff there. Isn't there like more plant
there
diversity?
is an orchid. Yeah.
I think there's more, because I saw it in a book once. There's a book in our library.
Okay.
I read it once.
It's a fascinating island. But yeah, Heard and McDonald have weevils.
Same
and,
species, different species.
Different species. So yeah, that would be amazing.
So it's
There's
cool
just
to go
so
to
many.
Crozet, because apparently there's a lot of invertebrates. And that's
Yeah.
weird, because it's very tiny islands. It's much tinier than Marion even.
Yeah, right.
I don't know why. Why would it be higher diversity? Is it just random? Has to be a
Island
reason.
age. This
Island
is sort
age,
of part of the
yes.
historical drivers that I'm trying to feed into my models.
I think
you
that's a
know.
good one, actually.
Island
age
age
is a big
distance
factor.
Distance
think.
from mainland or distance from an island that's been established for a long time. If they're relatively close, like the Kerguelen Plateau is very old.
It's old.
Very old.
I think it's sedimentary rocks there, even, if I'm not mistaken.
Yeah, I think it could be.
What did I
I need
read?
to check, but I know it's very old.
Oh, the South Georgia. No, sorry, the South Georgia that I read. That it was actually plate moving. It's not volcanic.
Yes,
It's
yes.
the plate movement, so
yes.
that's very old.
And South Georgia, had uplift and there's some interesting papers about there. Another island though that does not have weevils is South Georgia.
South Georgia.
Yeah.
And it had mice. They had a successful eradication there. That's weird.
It's really interesting. It makes me wonder, were there weevils that just never were detected in time?
And maybe the mice killed them all.
Yeah.
You
never know.
But that means
would have
they
thought
might
it
come
would be
back
found.
though, wouldn't you think?
Yeah, I would have thought. Well, I would have thought there'd be some maybe fossil evidence or something. There's been fossils found on some of the other islands of.
What is a weevil
there's actually
fossilized?
a weevil fossil that, a weevil species that is pretty much only known. Well, it's known well from a fossil, but there is one other specimen in a museum, but it hasn't been seen alive in the field for a long time. I would love to try and find that.
Yeah, when I came
Wouldn't
to
that be a
Marion,
great personal endeavour?
it will be. You should do it. If you can get the opportunity to get there, go for it. That's the one thing. Yeah, I think it's really cool.
Very fun.
I would love to go to another island, but I think I should just go back first and figure things out. And then I'll
No,
decide.
you're made for the islands, Elmar. This is you.
Oh, really?
Yeah, I've decided.
Oh, goodness. I like trees, Naomi. That's the problem.
I haven't seen the Elmar that likes trees. I've seen the Elmar that likes...
That's true. It's a different
that
Elmar.
likes the
This is the hectic, not Elmar. It's a different person. Gonna become someone else again when I get to mainland now. But yeah, I would love to go travel to see those islands as well. Actually.
Yeah, the New Zealand ones sound amazing as well. There's some really amazing invertebrates there. And birds.
What did they say today? Campbell Island was eradication program, right?
Yeah, they were talking about Campbell and...
Would that be a good island to go to then? To get some interesting results after eradication.
Yeah.
Actually.
Yeah.
That'd be cool because then
as well. Antipodes,
Antipodes, what is
yeah,
it?
they eradicated as well.
Oh yes.
That would be interesting. Like some of those, you're right, would be really interesting to see how recovery's going.
But I don't know how your funding works. Can you actually?
I think I'm definitely. I'm dreaming now.
Dreaming now.
Maybe
you never know
in the future.
Maybe Stephen will be happy to do it.
Sometimes the right project comes along, but that's okay.
Okay.
I'm happy to read about them for now.
How long do you think this PhD will take you? Do you think it will take you a normal time or more?
Yeah.
Just because of all the traveling?
I think it'll be at least the 3.5 years. Yeah. There's a lot of work.
There's a lot of work.
I've got, this is just one of many, many chapters that I'm working on.
Exactly.
To try and answer all these questions. So yeah, I've got a lot of work ahead of me still.
Yeah, that's cool.
It's scary to think how much, but I'm sure it'll all pan out nicely. Yeah.
I wanted to ask you, your husband, does he enjoy the weevils? Does he enjoy I don't know, all the outgoing things?
Yeah, I don't know what his interest is on that. Well, I think he's interested in my work. But definitely doesn't have the same passion for the weevils that I have. I know that he has to answer questions when he tells people what I work on. And, he'll say, my wife is studying weevils. And I'll be like, weevils? Is that what you find in your flour in the cupboard?
Yeah, that's what probably people would say. Oh, weevils, those pests on some of the fruit or whatever. You just get so many pests on food.
What's she doing with those?
Yeah.
Yes.
Throw them away.
Yeah. So he's probably more knowledgeable on weevils than he would have ever thought he was going to be. Yes.
and a very special weevil, because we talk about weevils, we talk about a lot of things actually in general, because there's so many species of weevils. I think it's the most.
It's very diverse.
Isn't it the most diverse family of animals in the world? I have a feeling it is.
Yeah. Well, yeah, beetles
I know,
in general.
Yeah, I know Coleoptera, the order is.
That's it.
But that might be the most diverse family, I think.
I did have that fact somewhere locked away in my mind that I've forgotten.
I also forgotten
it. Definitely very diverse. We can confidently say that.
Yeah, because I technically also worked with weevils before I came here. My master's was bark and ambrosia beetles, but they're also in Circulionidae.
Yeah, they
they're
are.
a sub-Xyloborini tribe. All the border. Well, there's actually a lot of tribes. Xyloborini is the one I was focused on. There's so many tribes.
Yeah.
There's this app. I don't know if you would enjoy it. It's called, what is it? Eruditus, something like that. I should actually check if it still works. But this is lab. It's the university. What university is it? What place is that again? It's in Florida.
Yeah.
Damn, can't remember the university's name now. But the guy, Yuri Holker, he did some ambrosia and bark beetles. He made this app of all the beetles you get. I can remember meeting these people. They came to South Africa for like a conference, and we went out to Richards Bay area there in Natal, and we just looked for whatever we can find, and different hosts, different tree hosts, different, shrubs, whatever. Just try and look inside of wood, look for holes. I've never seen people so nerdy about it in my life. I was like, I don't think that's me. I don't think I'm that passionate that I'll sit the whole day for 10 hours going for every hole. It was crazy, but I was just so excited and passionate about it. So
did they find any new species?
They did, actually.
Yeah. See, this is what keeps you coming back.
Yeah, they found very interesting species in the wood, and South Africa has no knowledge on. We don't have an expert on that. You need an expert on it, because we have a lot of tree species in South Africa, and you get obviously some that are polyphagous. I know of one in the forest that I worked in. There's an indigenous polyphagous one. Eats a lot of hosts, and then I found like a couple on certain species. You can see they're host dependent, and that's what makes it very interesting, I feel. There's so much to be learned. Yeah, when you drink a beer at night, like just before, because ambrosia beetles and bark beetles, they fly when it's no wind. That's why I actually looked here for bark and ambrosia. It won't be, there's too much wind here, and they need something to bore into.
Yes.
But just before sunset, and there should be no wind and it should be hot, you'll always see them fly, and they're attracted to alcohol. So when you drink a beer or you drink a wine, you'll see them fly around. So if you ever notice like a flying thing while having a sundowner, it's probably ambrosia or a bark beetle.
Yeah, right.
And not a fly.
Why would they be attracted to?
Stress. So sometimes tree
like
stress. They release like alcohol and all kinds of different defense mechanisms.
Right.
Yeah, so they're attracted to that, and then they go to the host plant because of that. Yeah, it's amazing actually. So you make an ethanol trap basically. You hang it up at a tree, but you shouldn't hang it up in a tree, because if you hang it on a tree, they'll actually go into the tree and not into the trap, because they think it's a tree.
Oh, it's the tree that's doing the calling.
Yeah.
It's
very interesting. So you should put it on a pole near a tree rather, or something, and then you can catch many.
Huh.
Yeah, it's cool. Yeah.
About
Just
knowing
to give
their
you some
biology.
history.
It's about knowing their biology, right?
Yes.
Yeah.
It's basically just to give you some history on some stuff I did. Weevils. Other type of weevil.
Yeah. There's so many types of weevils, but
crazy.
Yeah. That's really cool. That's really interesting.
Yeah, it's super diverse.
Yeah.
But yeah.
Nice.
So what's after this now? You're gonna go back to Australia?
Yeah, heading back. I'll be back in the lab.
In the lab.
Again.
Just measuring,
processing, and I've got other work on other chapters as well. So there'll be a lot of reading.
A lot of reading and introduction and all that kinds of stuff.
And also, and modelling with
modelling.
Get out some of these like climate
And you're
drivers,
using like
and I'm
old.
gonna have to put it into.
What data are you using for all that modeling? Is it like old data or?
I'm trying to get the most up to date, which is that's the hard thing. So some of it I'll need to be using like satellite imagery for temperature and wind and precipitation and it's going to be. There's a lot of work to do.
Okay.
On the computer in the lab. It's been lovely to have some fieldwork time.
So is it just this chapter that's going to be field work, or is the other mainly focused on monitoring climates?
No, there's some others that'll have potential for field work, but we'll wait and see
Wait and
comes.
see if we can.
If not, this one's going to keep me busy enough anyway. Yeah, but I'll let you know what I find.
Please
Yes.
keep me in touch. We should keep in touch and just talk about it.
You'll be able to find my results and read about it.
I'll make a comment.
Make a comment.
Oh no, Naomi didn't call. She didn't get enough weevils. She's gonna have
I
more.
was going to say, please don't make negative.
No, I'm just kidding. I won't do that, obviously.
Yeah, that would be fun.
Yeah,
Yeah.
great.
Yeah, well, it's been super fun.
Yeah, it's been super fun. Actually, I've enjoyed your company. It's just nice having people that's also into plants and into weevils, into invertebrates in general. Because the whole year I was with people who's not into that. You know, they're into other things. They're passionate about nature, but they're passionate about other section.
Yeah.
And I learned a lot from that because I also became passionate in everything then.
Yeah.
Now I also like the birds, and I also like the orcas, and I also like the seals. You know, now I like everything as well. So it's very interesting. It's nice to have passionate people. Like their work.
Yeah.
It just makes everything easier.
It's fun when you are in the field with people that are doing a similar skill than you, though, because the moment that we were all together trying to do the one task, it was phenomenally easier.
It was really easier.
Life just got a lot easier the moment we were all focused on the one task.
That's
so
true.
the minute you have even just one helper, it makes such a difference.
It makes
doesn't
a difference.
I mean, you can always do it alone, but it's always easier with someone else. that's
I enjoyed it a lot more with other people. Like I enjoyed being alone, but sometimes you just need another presence to keep you going. Sometimes, like sometimes when you're alone, you're like, I've had enough now. But then you see other person's energy, like, and then you're like, I also want that energy.
Or they notice something different.
Or they notice
like
something different.
Like we were talking about, you notice something, it's like, suddenly, your whole perspective shifts within a moment.
Yeah, a lot of people, like, I've noticed a lot of things about the moss, but it's always nice to take people who don't even know anything about moss, for instance, and they walk with you and they're like, so what's this one? No, that's that. And then you get there, but this one looks like that. I'm like, I haven't actually noticed that, but that person noticed it, for instance.
Yeah.
Yeah. So it's interesting. That's another cool thing I would want to look at, but I've looked at it briefly. Like the community composition of all the mosses, like all the micro arthropods or microinvertebrates rather, that live in the moss. And I've definitely seen a trend when it's dry, they go into the soil, they leave the moss. So it's like a migration thing between moss and soil. You can clearly see that was happening.
And did you note when you were doing your course, did you note the type of vegetation and moss that was on top of each?
So those cores
to match that sample.
Oh, yes.
So like you could later on do that work where you could be like, this community of mosses supported.
So for the soil course, no, I didn't do that, but I did do it for the one study. Was specifically on certain habitats. Yeah. So that one, yes, but not for the index trapping. That I didn't take note what plants grew on top of the soil core.
Okay.
Which would have been cool, but sometimes there's a lot, so it's difficult to.
Yeah, and also I
you
guess
don't know,
the amount of migration in and out of an area, you don't know.
You don't know,
like,
so
it potentially is not.
I actually thought about that as well, but then I just left it rather. I think
there's a lot to take. Like,
there's too many things.
Yeah, too many things you could monitor.
Monitor, yeah. But that's why I did like a different moss vegetation. And just to compare that, and I usually focused on the dominant moss in the area. So I won't take like an obscure, rare moss growing under a rock and it's just this little bit. There's no point in that. Let's just take all the general moss dominant in the area and see how they differ between altitudes and different habitats.
Yeah.
It's quite interesting. And
How
obviously,
many did you find?
like species.
Do you know? Yeah. Do you remember a count? Or did you do a count? Or
I can't give you an exact
50
number.
plus, 60
No,
plus,
that's
100 plus?
definitely more than 60.
Really?
I found at least 80.
Wow.
I think so. But I need to go look into it. Obviously, I do all of them. I'll have some doubles. Like how I sampled, I tried at the start. I tried to do like let's say I take like a 10 by 10 meter, and I just collect all the moss inside of that. That's like how I started. And then I'll find like, let's say 5, for instance. And I'll do it again. And I'll look. But if I can counter one that I've already sampled, I'll just sample a new one. So that's how I just build it up, and I just went around the island everywhere. Sometimes I didn't find any new ones. But if I think I found one and I'm not sure, I just collected it in any case. So yeah, I still have to look at all of that to see if there's how many exactly, but there's definitely more than 80. Definitely.
There's gonna be a lot of work in that.
Yeah. But anyway, I will have to come back for that. If I
Will you come back?
I'll come back for moss. If I can ID them now and get all the resources in place and learn more about it, how to ID them here in the mainland, then I'll definitely go back and go collect all of them.
Okay.
Like for the takeover or something. And then keep a collection there for the people there and for collection back home. Yeah, we need to do that. I want to do that because I'm quite pissed off actually at the base for you get there and there's no moss reference collection, but it's the most
nobody's done
it's the most dominant plant there. What's the point?
It's been waiting for you to come and do the work.
Yeah, but it's sad because Nick Gremmen was the guy. He was the moss guy.
Okay.
And he died unfortunately in 2019, I think.
Maybe you can access some old works that
he probably has a collection somewhere. And
true.
He has a 1970s collection there. It's super old, obviously. That's a bit old. And the stuff, the mosses were like breaking apart. But I used that. I used some of that reference to ID some of the moss.
That's cool.
But the moss names have changed a lot. So I don't know which is which because now I've used like old names. It's probably not applicable anymore.
Oh, you've matched it. Yeah.
But you can figure it
You
out.
can figure it out. Yeah.
So yeah, that was cool.
Nice.
I kept myself busy like that just for fun as well.
Well, it's a challenge.
Yeah, it's a challenge.
Naming all of those or identifying.
Moss is not easy to identify.
No,
And
I think you've
noticed-
said why, because nobody's done that work yet.
Yeah, no one's done it, and you need the microscope as well for some of them, because some of them are super similar, and there's no way with the naked eye to tell that this one is different. Same, especially if it's the same genus.
You're doing like slide samples with those.
Yeah, you need slides. I didn't make slide samples.
Okay.
It's the thing. I knew about it, but I was like, I can't do this work now. It's just too much.
There's a lot involved.
Too much.
Yeah.
I need to focus on the stuff Michelle obviously gave me first. That was like a side project.
Yeah.
That became
There's
like
only
my
so
life.
much side time.
Yeah, exactly. That's the problem. But yeah, it was cool. So yeah, I'll definitely go back. Do you think you'll go back?
I would love to go back. I'd go back if they turn the ship around right now.
That was too short. We needed another week of takeover.
I would have had a great. Yeah, I would happily go back. I think there's so much to do and learn and continue with. So yeah, I would love to. But
do you
who
think
knows?
you'll work like on other weevils in Australia after your PhD? Like, you'll do some postdocs, or do you think you'll stay at the cold?
I would love to do postdoc. I always love cold climate. I think I'll always be doing something cold climate, whether it be alpine or sub-Antarctic. Though sub-Antarctic has my heart now, so maybe. Yeah, we'll see. See what comes up. Yeah.
Okay.
Yeah. Well,
Yeah.
thank you.